Okay, so first of all, thank you so much. It's a real pleasure to be here. I'm a huge admirer of Vadim work, and we've for many years, and actually what you guys have been doing here, and so it's just fantastic to be here and to be able to discuss all of this in person. And so yeah, so what I'm going to do is I guess this will work, I guess, for you guys. To discuss mostly unpublished work and to throw out some ideas, but also sort of that I think reflects sort of the amazing kind of initiative and interfaces that you guys are at here, basically in terms of physics, engineering, chromatin biology, and also cancer biology, which is really very much at my heart. So kind of as Vadim said, as I said, for the graduate students, a lot of people will call me an interdisciplinary scientist these days, but mostly my graduate and postdoc advisors just sort of suggested that I lack focus. But now I'm interdisciplinary, but I just lack focus. But the overall goal of my research is really to identify critical growth regulatory targets and to try and really use this knowledge to impact and improve patients' lives, right? And that would be all of our goals. And viruses are just really sort of, how does a small genome take over a cell, right? And they're very clever. I'm not as clever, so it's sort of follow the virus. And they actually impinge on sort of the critical circuits that drive growth deregulations, the same processes, and they converge basically on these circuits. And that led to really the identification of many of the key targets and sort of continue to this day. So in terms of sort of understanding these targets, well, structure determines function. I was originally trained as a biochemist. And one of the key kind of sorts of challenges is really trying to understand structure in situ. And that required basically the development of sort of new technologies. And I'm going to actually speak mostly to that today. And then really kind of, I think the key challenge for us all is really, can we sort of leverage this knowledge, right, of these critical targets and structures to actually sort of realize the promise of genomic medicine, to redesign basically cells themselves and even viruses that can selectively seek and destroy them, okay? And that's actually sort of where my story began is, I was actually going to work with Jim Allison basically on the checkpoint inhibitors. This is around 2000, but I felt, well, that would be done, but could we actually find a virus that replicates in a tumor? And wouldn't that be sort of the perfect target to combine with immunotherapy? That's what led me to Frank McCormick's lab, who was sort of where the first oncolytic virus was being tested in clinical trials to target the loss of the tumor suppressor P53. I thought it was a brilliant idea, I still do, but actually we didn't understand enough. And the reason it didn't work was there was another protein, which I then discovered in my lab in Salk, where I went to learn virology, which actually inactivated P53. So we never had that virus. And then this virus actually is what led me to the genome, and that it actually silenced DNA in a way that just couldn't be explained by the textbooks. And that led us to questions then about, well, actually, what is sort of the structure of DNA in the nucleus? And if we can understand that, again, can we begin to understand and realize the promise of genetic medicine? So it's fantastic to be returning to sort of the more fundamentals today, because hopefully we'll be in patients, actually, which has taken me kind of away from some of this for the last 2 years. With first in human trials with some of the viruses that we've actually developed sort of in a company we span out basically for that. So now I'm back to the basic biology, and that's what I'm going to talk about today. So the DNA double helix, right, sort of provided basically the structural basis for how our genetic information is both copied and stored. But it's the structure of DNA in the nucleus that determines if it can be accessed and used, okay? But you can't crystallize a gene, right? And so that's the problem, right? How do we actually understand the structure of DNA in the nucleus, where you have basically sort of meter of DNA, more and more of DNA basically packed into a 10 micrometer nucleus. And really understanding this structure is fundamental to understanding basically the function of genes and the genome. And so, when I sort of was looking at the textbook models, beautiful work, by Olin and Olins, sort of with in vitro reconstituted DNA. Thank you so much. Sort of led to the idea that there was it was wrapped around nucleosomes, basically. And then again, based on in vitro modeling. advance it again. Not sure if something has changed. I guess I can control it from here, maybe. Okay, back. Okay. Let's just sort of the classic textbook model that sort of formed these hierarchical structures. But there's a reason it's a cartoon model, because none of these structures had been visualized in situ. In the meantime, sort of genomic sort of biology proceeded apace. And we know that basically DNA can be modified, wrapped around many different histone variants in nucleosomes. These are modified, and these can form sort of into these three-dimensional domains, TADs, LADs, basically nuclear bodies, all the way up to this. And the issue is that this spans multiple scales. And each sort of technique can sort of interrogate one of them, right? Again, it was a sort of both Feynman and Francis Crick said various versions of, if you want to sort of understand how something works, just look at it, right? But easier said than done, because again, we can sort of look at these as sort of individual scales. But what we really need to understand is how they're integrated inside the nucleus to give rise to function, right? You know, with quantum computing, maybe we can take all these different dimensions and begin to understand it. But the cell does that actually by integrating structure in the nucleus. So what is the structure of DNA in the nucleus across these scales? And if we can understand that, can we begin to understand the basis of sorts of cell fate and actually disease? And that's what really led us to develop sort of chromium T, which I think sort of together with these other techniques helps fill a gap. And sort of it was actually a fairly simple idea, which was really, you know, is there a DAPI for DNA? Because the problem was contrast, right? How do you create selective contrast of DNA in the nucleus to make it electron dense so that we can sort of visualize its structure, right? This is a silly video, my favorite movies. This is Indiana Jones. And this is sort of The Last Crusade. And he's kind of going over this bridge, which seems to disappear into the background. And he has this trick. And if you understand this, you'll understand everything. Okay. So he basically throws dust over the surface, right? And so that it now can become visible to everyone else. And that's basically what we did, okay? That's fundamentally it, right? So how do we do that? To create basically a surface map of DNA and chromatin in the nucleus. So certain inorganic dyes, and actually just in molecules in general, have a really interesting property, which is that when you excite them, in addition to emitting a photon to go back to the ground state, they can actually undergo something that's called intersystem crossing and donate an electron to a higher, basically, orbit. And in the cell, that can actually be sort of donated to triplet oxygen to create highly reactive local singlet oxygen, right? So it produces basically this reactive oxygen right around it. And so, which can actually then be used to catalyze the deposition of that Indiana Jones trick across basically anything that's intimately associated with DNA within a few nanometers. So we screened for dyes, and we found one, DRAC5, a membrane-permeable DNA binding dye that binds to DNA. And what happens is you'll see us switching on the light here, basically. And in the presence of this other small molecule, DAB, that's the dust, it actually catalyzes it basically into a polymer that sort of sprinkles down on DNA and associated proteins. Let me be kind of, again, very clear what this is and what it's not, right? It's using DNA, but it's creating a surface map of things that are intimately associated with it, which can be nucleosomes and other closely interacting proteins, right? And so that's what's kind of interesting about it. You're not going to see helices or anything like that, but you will actually begin to understand sort of the general densities. And this polymer basically then, DAB polymer, is osmiophilic basically. So it binds to osmium, the heavy metal, which can deflect electrons and allow us to visualize it in the electron microscope. And this is all together with Mark Ellison's lab. So osmium basically binds to alkene groups. And so this is the classic sort of a TEM, a thin section. And you can see the membrane stained, and here's the nuclear membrane. And that was a cell kind of half in our field, just so you understand what you're going to look at in the rest of the talk. And you can see suddenly that the DNA sort of is black in here. But the problem with a thin section is that it's 17 nanometers, so you're projecting through this massive volume, and yet things we want to see are 2 nanometers and 10 nanometers, right? And that's, again, now where multi-tilt tomography comes in, right? And so basically, And obviously there are probably way better algorithms as well for doing this. But we initially used sort of 8-tilt, multi-tilt tomography, which basically sort of effectively creates computational slices, which are about 1 to 2 nanometers. And then you can actually travel through the whole volume. And sort of the classic picture, this is of the nucleus, not dissimilar to anything you might have seen in the textbooks. When we take 80 slices, this was a joyful day, sort of resolved actually into this. You can see the gap in the membrane here because nothing sort of is stained. And these little black pieces in here are actually nucleosomes, basically, so they're consistent with the volume of the nucleosome, and an individual sort of free strands of DNA, right? But then joy turned to horror because, you know, as I said, I really was coming from a very different area. We really couldn't see these sort of higher order fibers, 30 nanometer, 120 nanometer fibers. Everything sort of fluctuated in a way I would call it a heteromorphic polymer between about 5 to 24 nanometers, basically. And so at that point, then we thought, well, let's look at the mitotic chromosomes. This is obviously where it's most condensed, you know, sort of the sort of iconic image of the human genome, and yet actually also a black box, 50% protein, 50% DNA. But could we now peer into the depths if this stain sort of had sufficient resolution? And so, and I'm moving quickly through this because this is published, but just to give you a sense of where we're kind of coming from this. And what you can see was very beautiful. It's more densely packed together. But again, the sort of primary polymer structure per se did not necessarily change, right? So again, it was roughly between 5 to 24 nanometers. And I think that's kind of interesting, right? Because effectively, you know, if structure determines function, if there are only a few structures, then how can we actually achieve many different functions and all the different transcriptional outputs? I mean, look around this room, every single one of us is different and has a different phenotype. And yet, basically, the cell cycle occurs in the same way, right? Development has occurred in the same way. I mean, that's remarkable, right? And I think that's so exciting, right? And basically, this comes again to the idea of sort of critical phenomenon and how things can come together actually to dictate sort of key processes, but also give rise to variety, basically. And disorder can give rise actually to order as well as chaos. So when you have a flexible and disordered chromatin chain, then it allows actually for many different structures, which can explain potentially the different outputs that one could have, and many different histone variants and things like that. It also explains potentially if the primary polymer structure doesn't change as you go through mitosis, how that information can be inherited from one part to the other. Because if you change structure, what is remembered basically? And so these can be inherited because fundamentally it doesn't change. But now the rest of this is actually unpublished. So now the question is, well, that's all very well, but that 30 nanometer fiber and these 120 nanometer fibers were kind of comforting because they kind of provided a natural model for how RNA polymerase might be excluded from basically the DNA to be able to transcribe the genes. But if everything is the same sort of diameter, I mean, now what, right? How do we basically explain that? So we think about sort of DNA, polymer is actually kind of a misnomer in ways because it's actually not really made up of a repeating unit. Sequence is actually kind of different in a way. Each nucleosome can be different, each of these kind of compositions. So in that sense, it's heteromorphic, okay? Second, right, you know, there's different models basically where H1 sort of tail interactions can mediate interactions between chains, which I think are very elegant. Protein nucleosome bridges, and you'll hear a lot as well about liquid-liquid sort of phase transition. And the thing is it one or the other, or how do these all act together? Is there sort of a fundamental underlying principle, right, that has to be simple, that can actually sort of explain that? So biology is always sort of, I think, best understood in the simple language of both chemistry and physics, with fundamental laws it must actually obey. And so I'll come from, again, a biochemistry sort of background, but it's been it echoes very much actually some of the ideas that Vadim and others have put out there as well. So it's a really interesting phenomenon that basically chemical and polymer scientists have been using for years in the plastics industry, which is that certain flexible, linear flexible polymers basically undergo what is called liquid gel. And I'm not talking about liquid-liquid, I'm talking about liquid gel state transitions at a critical concentration, which in a good solute is at about 20 to 30% vol-vol. And so effectively, as they approach the critical overlap or entanglement concentration, effectively they become entangled. So imagine you're in a tangle line and you can't actually sort of get away from each other, right? You all start to get enmeshed basically, and you kind of begin to move as a mass basically in a crowd, right? And so you begin to undergo sort of these, so you can merge basically from liquid to gel, and these can become actually volume excluding and have really interesting properties. So what in some ways, if it's simple, right? Because, you know, if we all have different DNA sequences and different polymorphisms, and basically we can trace trajectories back to yeast and all these different organisms, what's the fundamental principle? How do we build on a fundamental principle other protein apparatuses that could actually sort of give rise actually to the fundamental phenomenon, but have different kinetics and regulatory mechanisms? And so one question then is, maybe these different mechanisms I just described in the previous slide, what if they actually just mediate sort of chromatin concentration, basically? They can drive chromatin concentration in different ways so that it can approach a critical volume, which would exclude basically the transcriptional apparatus, okay? So how do we do that? Well, the nuclear membrane and basically the lamina-associated domains are known to be silent heterochromatin domains. So why don't we just take a look basically at them then, because we can do that. And this is the power of the tomography. So you're going from one side of the nuclear pore now to the other. You can see the chromatin sort of associated with the nuclear membrane, see the loop as it goes around here. But it's kind of only when we started to render this that, you know, begin to understand it. And this is sort of what you would see if you were a transcription factor kind of coming into the nucleus or a virus. Sort of reminded me of kind of a kelp forest, actually, you know, sort of, and this is again this A-B compartments where basically if 40% of the genome is there, begins to channel you sort of in towards the center, right? And sort of a discontinuous gel filtration column, what I might call it. And so what is the concentration then basically at the nuclear membrane? And so what we did, again, these are just surface rendered maps of the chromatin through slices. And you can kind of put basically an overlapping sphere because I'm a climber, basically, and contour maps make sense to me. And so basically it was like, can we create topographic, 3D topographic maps of the nucleus of the different volume concentration densities? So this is actually where we know there's this heterochromatin up here. And what's interesting is they really have very different concentrations. So this is around 40%. And then in towards the center, you're getting sort of in around here. So it's about, this is a somatic primary cell. It goes from about 20 to 45%. And here's mitosis. And you know that the chromosome at mitosis must move as a gel. That's the only way it can resist drag across basically the nuclear plate, okay? And so effectively, this is a really high concentration. What was fascinating to us is it's exactly sort of the same concentration, right? About 47% as that at the nuclear lamina, okay? Where again, that's the concentration where it can undergo a state transition and where it's transcription inactive. So what if it's that? And so that's sort of one of the, so we kind of come up, you know, this idea of a chromatin volume concentration, and I'll keep it sort of at that. There are other parameters as well, but we could do with a lot of help modeling this. We're not physicists. But effectively, you can render this in 3D. And sort of, you know, silent heterochromatin can exist up here, and you kind of have this kind of continuum. And then basically mitotic chromosomes are here. And so what we've been doing basically is sort of then looking at basically stem cells at different states of differentiation to create maps to understand sort of what those look like. Constitutive heterochromatin, such as effectively chromocenters, polycomb bodies and things like that, epigenetic drugs. But today, given your engineering focus, I'm going to talk actually about, you know, an individual piece of DNA that goes from the silent to the active state. So the principles kind of come from that. So the issues with what I've shown you is that, you know, only a few percent, you know, basically about 4% of the genome is coding. So what am I looking at really, right? You know, that's the problem, right? What can I say it's active or not? So I really wanted to see a gene. I've always wanted to see a gene, okay? And with light, we can begin to sort of, this is Bo Wang's paper, you know, with Cas9, we can sort of look at its localization and position. And we could, you know, sort of use basically a correlated light and electron microscopy to zoom in, but you kind of have a fine, you know, where's Waldo problem, where it's like, okay, but where's the gene, right? And so it's sort of structures that what we really needed was effectively kind of a two-color EM. I needed a genetically encoded fluorescent metal nanoparticle, because we discovered that actually once you permeabilize the cells, which explains sort of some of the 13 nanometer and other structures, you really sort of disrupt structure. And so the thing that you see in the electron microscope is densities and metals. So how do I create light with metal and density? And the answer to that is sort of to look to nature, and that we actually store metals in all of our cells across all kingdoms of life in a protein cage, which is made up by ferritin, which is a monomer. What this thing kind of does is it basically assembles as a 24mer into this beautiful octahedron cage, which reminded me of a virus. It permeates my thinking. And acts as a cage which can store 4,000 iron atoms in the center, which is naturally electron dense and can be seen in cryo. So I thought, well, what if we could take this and actually now sort of reimagine it, actually, and re-engineer it so that it actually can be a genetically encoded probe that would store iron in the center so I can see it, like a genetically encoded nanogold. But now I want to put things on the surface so it can find genes, and so it is fluorescent, okay? And that was very hard. And we nearly gave up. This was Gavin, because there have been several papers that have been published on this. And I think what they're seeing is quasi-equivalency, where they see steric hindrance because, you know, we were not successful in seeing them in the electron microscope. And the reason was, this is a native gel when we were 3 years in and almost about to give up, where we took E. coli ferritin, and this is a native gel. It should run up here if it's a cage, but it doesn't. It doesn't assemble in mammalian cells, and then it doesn't store iron. And so then we just looked across kingdoms of life, and horses, for courses, for some reason, that actually assembles beautifully in the nucleus of mammalian cells. And then it can store iron, so we're like, yes. But now we needed to synthetically engineer it and create something new. So we took green fluorescent proteins and nanobodies from llamas and things like that, put them all together. Mostly we got a mess until about 300 goes in. And this was really, again, somewhat structure-based design and a little bit of directed evolution to get something that would assemble a proper cage at the top of a gel. So what the idea is then, we call this fire nano for fluorescent iron EM nanoparticle. And you can kind of put a nanobody or anything you want here, actually, really. A fluorescent protein, and then basically this is your iron cage. So you can genetically express that in cells, and it can go to the nucleus if you want with an NLS. And the idea was like, if you have a Cas9 targeted gene or locus, it would actually then find it, right? And actually give you a mark, that specific DNA sequence, okay? So before, with any unknown technology, you should test it against a known. And so to do that, we looked to this beautiful system developed by David Spector, which has been fundamental, and Rob Singer and others, to really sort of understanding the dynamics of RNA export and transcription and things like that. And what it is if there's 200 copies, I want to be very clear, there's 200 copies of a transgene which are integrated into a single locus, 1p36. And these have basically lac repeats, which can be bound by lac GFP. So that allows you to see a spot. And there you go. But then basically, so we basically made a fire nano, which could bind to GFP, but was red. So there you go. We're very happy with that. Forms a nice spot. And what's really cool about this locus is that it's actually sort of facultative heterochromatin. But in the presence of doxycycline, it has a teton promoter. It turns on a CFP peroxisome protein and an RNA, which has MS2 transcripts, and which become important. We wanted to make sure we weren't perturbing transcription with our new device that we'd engineered. And so what you'll see us now doing is add doxycycline, and the fire nano and lac GFP is there, kind of expands a little bit as previously reported. And you can see that you turn on perfectly the peroxisomal protein in the cytoplasm. So we felt pretty good about that because, again, key Heisenberg uncertainty principle was like, you always do not want to basically sort of disturbing the very measurement of the thing that you want to learn. Okay. Now, what's cool about this is that this locus is actually facultative heterochromatin. So when it goes, so in the presence of doxycycline, basically it has HP1 beta initially, sort of that will dissipate, and it's also encoded, encased in a PMO body, which is great interest to me as a virus as well. And then again, sort of that begins to open up. So now we can do something really cool, which is iron has a different density, sort of in the periodic table than osmium. We can kind of have two color EM now, effectively, right? Where we basically stain the chromatin with our chrome EMT, okay? But now, basically, could we see individual nanoparticles and find basically the actual gene structure and visualize a gene in the active and inactive state for the very first time? So that's what we did. We took the fire nano plus or minus dox, and then we did chrome EMT, okay? So, and here's the silence locus. Now, remember, there's 200 copies of the transgene, right, integrated into the one place, okay? I want to be clear about that. And each of these dots, took so long to work. I thought it was April Fool. I thought it was a Sharpie marker at the time. They were just playing with me, but it's not. So I've gone into a single slice now. So each one of these is actually your 8 nanometer iron core, basically, and it marks actually a lac GFP that's bound to the DNA. So you're actually seeing sort of individual sequences and the DNA. And this isn't the silent case. So now what we can do is we can effectively mark sort of lac sequences through this, both fluorescent band here. We're taking a trip through it. And I'll just turn up the contrast view so you can see it. And then I'll show you what it all sort of begins to mean. Oops, sorry. Go back a second. Yeah. And so I'll color in red now, basically the fire nanoparticles for you. Sorry. Manually. There we go. There they are, right? And so now we can see all this, and there's the chromatin. You start to begin to see this haze. If you look at it long enough, you'll see the Death Star. And so basically it forms this remarkable sort of a ball, right? And so now the question is, what happens when it's active, right? Same sequence, same everything. What happens? Does it change structure? And by the way, here's the scale bar. These are not higher order fibers, okay? You can see that for yourself, right? So here's the active locus, and there's a whole ton of things that I want to discuss with you here. So let me just play a few first. So now basically we've turned it on. And people had thought that the expansion of the locus initially reflected sort of an unfolding of a hierarchical fiber. Actually, it reflects a change in actually the concentration volume and actually the three-dimensional organization and the interchromatin distances actually between the polymers actually. And so now basically this is the lac sequence, and there's some really interesting other things that are going on, okay? But let me just take the first question. So is there basically higher order fibers basically in here, right? So here it is, and we can now just measure through the entire sort of volume. In general, basically, the answer is no. Again, mostly it's 5 to 24 nanometers, where you actually start to see, I'll show you active transcription, you'll actually see an unfurling, but for the most part, again, we're not going from these hierarchical fibers into actually sort of that the primary polymer structure is mostly disordered or heteromorphic, okay? But there are beautiful branches and structures. But there's something really super cool that we can do is that about 40% of the genome is almost viral, right? It's actually repetitive. But how do you actually see repetitive gene sequences, even by genome engineering or not? What we can actually do is sort of statistical clustering, because I know that each lac transgene has 256 lac repeats. And so I can actually identify individual genes because I have 1 nanometer isotropic resolution, okay? And so when you do that, you suddenly see the lac sequences cluster here. And so they begin to define actually each individual transgenes. We can actually begin now to see the 3D organization of a repetitive locus, right? So now I'm here, now I'm projecting basically the chromatin structure underneath it. And then I can basically, by inference, here's the other sequences basically, here's the tetramotor, here's these other sequences. And I can begin to put them together. And you'll see actually a dramatic sort of a rearrangement of these actually. And I'll try and explain it in the next piece. And so this is us modeling it here. So basically, this is the lac part of the gene, and this is the sort of the TET kind of operator. And it's amazing actually the way they're actually arranged basically into this this pole. It's actually it's quite uniform actually. But what happens, as I'll show you when it gets activated, is actually basically some of the TET genes, it actually gets sort of, it's not activated all at the same time, they don't all open up. They actually begin to open up at the outside, but that actually pulls basically on the other genes. And now you actually get this three-dimensional rearrangement of other genes that actually begin to go to the inside, which again, until I saw it, I was like, oh yeah, cool, right? That would actually have this effect in 3 dimensions, basically, where suddenly it starts to do this actually, right? And it starts to push some of these other sequences again into the center, which is really interesting in thinking about actually multi-copy genes, which are viral, they're translocations in cancer, and beginning to think actually about how actually these different mechanisms may play out. What's also really interesting is what happens is there's a critical change in chromatin concentration, where effectively this is basically a border at about 35%. And then actually what happens is you'll actually start to see this get into about 12 to 20%, but you'll actually see some denser cores beginning at the center because this becomes self-inactivating at the outset, right? So that's the critical change that's occurring, was actually the concentration starts to dissolve kind of from the outside in, and that can actually create basically individual structures on the inside. And that I think is very key for then understanding RNA polymerase. So here's Pol 2, and here's the locus here. And when it gets activated, you start to see RNA Pol2 beginning to gain access and to invade into it, right? So where's Pol2? Where's the RNA? Can we see that? Can we begin to see all of these elements? And so in the active locus, I said to you that there's lots of other things going on. Only in the active locus do we see basically the appearance of these 30 to 40 nanometer polymers, and these are just osmium densities. And these are actually big densities, which always occur at the periphery, basically. Only ever see it when you get activation. So it's a function of gene transcription. And these amazing factories that appear right at the periphery, basically, upon gene transcription. So what are these? And so one of the things is that could these be sort of mRNP assemblies or even RNA DNA hybrids, which I won't get into, but that actually could represent potentially, as you're getting massive amounts of RNA being transcribed, very different to a single copy gene. It's actually a central feature by the way of an oncogene, as well as actually viruses. Maybe again, this is what you detect is the lack, that change in homeostasis, because that concentration then drives basically these interesting densities. And now Rob, in his wisdom, had put an MS2 basically binding site into the RNA. So we just used MCP GFP, and we use Fire Nano now just to see the RNA, right? So we can use the Fire Nano now to mark basically the RNA, the GFP tagged RNA. And what's really, really cool is most of the MS2 starts to accumulate actually around these densities. The RNA is more accessible, so labeling, even though this is the same basic affinity for GFP, labeling density is greater. So each of these represents a single cluster. You get about 18. So you're seeing individual RNAs that actually start to accumulate and pile up in these amazing factories at the periphery that we can see at sort of this resolution. And then one of these particles basically, so here's sort of this amorphous density in between, which is very interesting actually. These are basically the MS2s, but these are the particles. And actually, when we actually superimpose them, the MRNPs are associated right on top of basically the Fire Nano associated RNA, right? So that's why we think that they're basically RNA associated sorts of particles, which may actually reflect either the spliceosome or the mRNP basically. But we can kind of get to that kind of a resolution now. Now, our first guess was hoping that these might actually be RNA polymerase, but they're not. And so the reason, so this is sort of, again, just to summarize where we're at, you go from dead star basically where it starts to open up the periphery. The RNAs get transcribed, high concentrations. They form condensates. And then actually you get sort of RNPs basically beginning to associate with the MS2 RNAs that occur there. The lac and tet basically fold out. And this is where you're starting to get basically the chromatin. Okay, what about RNA polymerase? So basically, we took GFP RNA polymerase. This is with Xavier Daszak. This is alpha-mannitine resistant. This is basically FRAP experiments. So you can, because there's 200 transgenes, you see RNA polymerase there. So can we see RNA polymerase on a gene for the first time, right? And that's interesting to us. So here's Fire Nano now seeing just the RPB GF RNA. RPB is the major subunit of RNA polymerase. Kind of interesting, again, to talk about protein-protein phase transitions. And obviously, being able to see the chromatin allows us to see the DNA polymer transitions as well. But can we now begin to sort of get a handle on both? This idea that at high concentrations of RNA polymerase, potentially as superenhancers and others, the CTD could associate through low affinity interactions to sort of drive sort of transcriptional initiation at a critical mass. Well, does it? We have 200 copies there. Where's RNA polymerase? So why don't we look? Okay. So here's again your chromatin, right? Basically in the density. And now basically each of these little black spots is actually an RNA polymerase. Okay. So let's so now that basically will actually mark basically the RNA polymerase, and you can see it basically sitting on top of the DNA. And it's actually several things that are really cool. But let's just take a trip through the locus. There's a little bit of a cluster here. I'll come back to it. But mostly actually they're uniformly distributed, which was actually very much predicted actually by Ibrahim's, Xavier's, Rob's sorts of experiments over the years in terms of actually sort of statistically predicting it. Every now and then, let me see if I can find you one. See these little two? They're right next to each other. We think those actually might be right on the promoter because they're individually associated with DNA. Some of these that are out here just are diffusing away. They're actually in the middle of space. But every now and then they're actually a cluster. But here's our 30 nanometer particles. I was hoping that that would bind to that. It's not. We're still trying to figure out what this is. But here is the RNA polymerase. So now we can actually begin to build these things in. What's cool is we can actually kind of ask a key question is, when do you get clusters of RNA polymers? Is there a critical number? Because we can actually count them. And it turns out, so far, basically, we can see 70, we can see 15. 15 is the minimal so far. But there's something really interesting that when we draw the distance between each of the RNA polymerases, it's not random, in fact. It's not at molecular distance either, but effectively the distribution always falls between about 15 to 30 nanometers. And the CTD of RNA polymerase is 16 nanometers, if stretched out, which is the least entropically favorable structure, of course, that it could adapt. But it's actually about 15 to 30 nanometers, which would be consistent potentially with the CTD mediated interaction. So we think that there might be a critical number such that we can measure, but also a critical distance that is consistent with it. I'll just end with sort of answering a key question, is that people will draw some of these sorts of protein condensates as kind of being driving, kind of binding into each other. But with the case of RNA polymerase, that would actually potentially inactivate it, right? Because it would actually take it away kind of from the DNA, right? It's supposed to actually zip along the DNA. But could it actually be then associated with chromatin? How could that work, right? Which is it, right? So we can actually answer that question because we can actually look. And so that's what you're seeing here. So we just took the yeast Pol2. And again, I'm not modeling, I'm not modeling helices before anyone screams at me. I'm just taking just the general density and shape to see if it's consistent. We can take each of these spots here and look at the chromatin and say, is it on chromatin or not? Okay. Or are they actually intimately associated with each other in 3D? And you'll see that here. So let's look at that cluster. This is very near to that other one. So we drew these in actually. What you'll see is actually that density was very gratifying. Is the fire nano was actually exactly the right distance away from that and the chromatin, which you'd expect because it's binding to it. What you can see is that these are actually associated with chromatin. So potentially it can actually stretch across potentially these structures as they mount up and mediate that with the CTD, and potentially these gossamer-like interactions perhaps can actually mediate these densities, which I kind of thought was a bit of a conundrum actually, right? So you can have potentially polymer-polymer interactions and protein-polymer, protein-protein interactions coexisting. And that may differ depending, again, on whether you're dealing with activation versus repression. So my last slide is just to sort of, two things I want to leave with you is there a general sort of a principle where chromatin volume concentration, which could be mediated through different devices, polymer, protein, and other interaction spatial, that could actually sort of determine whether it can be accessed by machines such as the spliceosome and other. This is sort of a somatic cell. This is our heterochromatic locus, where basically it's kind of going from here to the active. That's what I just showed you. So it's consistent with that model. We've also been looking at human embryonic stem cells and differentiated cells, and these sort of seem to be more generally open and sort of exist on this continuum. So, and also kind of polycomb bodies and other things. So right now it's generally consistent at this scale. I have no idea at a single gene level. I do want to be very careful to say that. At the scale, this is consistent and I think intriguing. This allows us now, I think, to really go after sorts of dynamic chromatin structures in a really interesting way. And so what you saw was basically sort of a multi-copy transgene. It gets activated actually from the outside in, and it starts to unfold kind of here. High concentration RNA basically drive then condensates at the periphery, and the association of these particles, whether these are activating or inactivating, remains to be determined. And you'll actually see RNA polymerase and some clusters sort of beginning to kind of pile up here. Then you also will rearrange the 3D chromatin structure of tandem genes and start to create some really interesting effects as you pull on them. And so it changes your ideas about how we begin to think of newly evolving genes, right? Because exonization drives gene structure and all these different things. I'll leave you with this final slide because I will, on an open call, is that, we have all these data sets, but how on earth we actually analyze them basically, computationally. I need help. I would love help because there's just so much information in them, and how we statistically describe this, I think, is obviously sort of right in Vadim's wheelhouse. So this is a completely sort of, selfish kind of call going, please help. But one thing I'll leave you with, because this is actually UTOS cells, which is an alt-driven cancer, a deadly cancer actually in children. And this is this lac locus that recombines basically, and this is again how you get amplicon. I just think this is really interesting. So that's this lac transgene now, and it's gone into another locus, and it's multi-copy basically. But what is amazing is here it is. Oops, just want to play it to. This is kind of a Death Star-like effect. Is that while this is not the same structure, right? There's actually more repeats in this, as what I just showed you. Even by eye, I can see that this is statistically similar, right? Again, it forms this kind of almost Death Star-like structure, becomes more inactive. There's actually more volume of DNA, harder to activate. And it really does remind me of Luke Skywalker, right? And it's more separated out basically. So the copy number has increased. And again, it's gone into this other locus, but at that particular sort of, I'm not sure if it's a critical genomic mass or in situ mass that can actually drive sort of now either polymer protein driven interactions that can actually lead to a similar sort of structure in a completely different place with potentially kind of a different context. And I think that's kind of interesting and really relevant for how viruses integrate, because they tend to do it as multi-copies. Amplicons, EGFR, glioblastoma, that's how we get drug resistance. And so again, can we begin to understand this? This is probably fundamentally driven by polymer and these protein low affinity interactions, to begin in my ultimate dream to realize sort of drugs for diseases that there's no hope for. So you're the hope. So with that, let me thank everyone who contributed in my lab. This is an amazing collaboration with Mark's lab. Get into the Fire Nano, Horn with the Chromium T, and yeah, I'm happy to take any questions, or I'll go to the panel at the end.